what we'd like to do today is to kind of work towards a roadmap. And we have an approach that we'd like to share with you and interact with you on. So when this meeting was being prepared, Chris Marshall in the ETRIX project came with an idea saying, hey, could we do some road mapping and so on. And as I was reflecting upon this, I realized when you make a trip and you plan a road map, you need a map. You need a map first, you need dimensions. And I was thinking, all these things are very valuable, but if we can in some way put them somewhere in a spatial relationship to one another. And so I reflected on some of the things I had been doing for OncoTrack and realized that some of those elements, these concepts, could be helpful in defining a number of dimensions. I would call them functional dimensions. So in fact, we're looking here from a functional point of view. What's the functions we want to have happen kind of independently from what is underlying, although we try to also be aware of that. And so what we'd like to do is explain you this. So what I did, I took such a functional, a number of dimensions and tried to every user requirement that I saw at a high level, place in that dimension, in these dimensions. And then I looked at different projects that I knew and tried to, based on the requirements of these projects, see where do requirements fit. So based on that, we have a kind of a straw man of things to think about. And what we'd like to do is, I'd like to run through the straw man with you as a first step. And then I'd like to break the group up in 3 subgroups who will each take part of what is being explained and reflect about it. See, do these things cover what we want? Do they reflect things correctly? And the idea is that the work groups come back after some discussion with a proposal of something that could be a means for us to say, okay, that kind of covers the main landscape of what different projects are about. And then in an exercise to check with it, for every project to indicate how much of a priority some of these functions are. So other data types are the standard proteomics, I presume, and I'm not so sure, but I presume that is available. When I put in 0, I mean I think it's available. Again,
what is standard proteomics?
That's a good question. So again, if you're in that group, correct it. I mean, correct it or yeah. What I did see is in one of the projects there was a loop proteomics type.
So for that example, I think that might be just break out. Maybe that the data management model is actually fine, but the loading scripts need to be significantly because the primary data formats are quite different.
Yeah.
So you have gaps at different stages of these pilots, which is why I want to do it completely different.
I'll offer myself to go around the groups, the breakout groups, to try and, as far as my knowledge goes, try and address what this is referring to, whether what we call standard proteomics, what tranSMART can currently do and sort of what it can't. So I'll be able to go around the table and help with these kind of things. Yeah. And anyone else with a technical understanding, please help.
Well, the bottom line, if it's a 0, right, what would happen is that the use case would come. So you can say, okay, ready use case or ready user story, but that's the story that actually exists.
Yeah.
And then it's very easy to say, is that the story that you want?
Yeah. So because my knowledge is limited, I put some things for verification, and that's one. What I saw is that there was a group who was asking, apparently there is mass cytometry, which allows for individual cells to be minimally measured as to proteomics. Apparently that's a request, not a high priority, but one that came in. The question that was also posed was, we have MS, we have HPLC. In fact, we have all kinds of multidimensional data that are not necessarily the ones that fit with the gene expression and so on. Could we be able to get these result types? Like for instance, you have a xenograph, a mouse model, and you have some results that come in different formats. Can we work with some kind of a generic multidimensional thing that we don't have to rebuild for every type again? And then there was the last one where people were saying, well, in fact, we have our own data format. Could we not just put this kind of, have a container that is kind of a black box where we put something in and we you know what type it is? We kind of pushed back on that one, but I think in a number of cases that might be someone. We pushed back, for instance, in the DNA with the OncoTrack people, and we said, well, couldn't you bring it in a VCF format? At least then you have a standard format, and then we can leverage all kinds of functionality that format associates to. So now we've talked about data models, or data models for the data part, but in fact, you're not only modeling the data part, you're also modeling the patient. And for instance, with a patient, we see again in OncoTrack that in a patient you're looking for how does he respond, typically an observational study based on the standard therapy, but you want to do more. And so what in OncoTrack they are doing, they're taking some of the tumor from a patient and implanting it in mouse or in animals, and then doing experimentation with that, which means you get a new subject because you want to follow that subject. There are some others where they will do, for instance, in vitro blood that you have, do some treatment on it, see what happens with that. So in fact, if you want to support these kinds of things, you have another data model object being the subject, which becomes more generic. It's not just a patient. It could be an animal. It could be in vitro. It could even be in silico data. And I think the tree that you have with the clinical data can work there, but I presume you will have completely different branching of these things, which on the modeling side probably is not a big deal, but on the visualization and searching side, it will require some extra attention. So that's another dimension. The third dimension is what we've captured all along, is the time dimension. So we are able to capture patient visits, but we're not able to say that patient visit was at that point in time. So that's another one that people have flagged up as being important. Go ahead.
In the sense that if you evaluate and you score 1, 2, 3, and you give it a 3 because you have a workaround in your own tool on the side.
What's that?
Let's assume you specify functionality
Yeah.
as being a 3 and not time pitfall.
Yeah.
It's there in the end because you have your own
Workaround. Yes. Yeah.
Would be there an option to say I have a workaround, and if people are interested, they can look for your workaround. So it's now more people are rather trying to score it in their own view.
Yeah.
And say it's not my problem, but I have a workaround. I do something else.
Yeah. Yeah.
That's not high priority.
Are you suggesting to put another code to say I have a workaround for that?
Yeah.
Yeah. Okay. Let's do that. Let's call that for maybe workaround or W for workaround. No, W maybe for workaround. Yeah, good suggestion. So I push on function and page down. Okay, and then there are a few more things before we go into the other dimension. So granularity in data structuring. So the more you want to dig in a structured way and querying things, the more granularity you need. So again there, the first thing could be, we were talking with the OncoTrack project and saying, okay, we want some DNA information to search for it. And I think I put as a 0 the fact that in fact, if you have some key information, SNP information or whatever that's important, you could feed it back as secondary data. Kind of, if the data is not too complicated, you could feed it as secondary data, and then it comes into your left hand of the tree, and so you can search by it. Because right now, searching cohort search, you cannot go into the multidimensional data. So that's one thing. another thing that we realized, yes?
Give an example of that, just to make it more clear.
For instance, we have a study in breast cancer where Veridex has developed a measure, kind of where they say, based on lots of genes, are you a high-risk patient for metastasis or a low-risk one? Well, that distinction could be something that you put in your left-hand tree and start searching on.
Yeah.
Yeah.
So there's a calculated field from other fields, something like that.
Yeah. Yeah. But it could be like, for instance, if you have a SNP variant that you say this is very important for us, you could kind of put in the left SNP A, SNP B, SNP C, and use that for your search criteria. Because you don't have, right now, you don't have the possibility to search based on your multidimensional data. A second possibility would be to say, okay, I can search on my multidimensional data. I can do a number of things, analyze a number of things, but basically what I want to do is consider tranSMART as this repository, and from this repository I extract information to an environment, I export it so that outside of tranSMART I can do all kinds of things. So, but if you want to do that, you need to have a way to identify that as an object that you want to export. And then the last level is, yes, I want to get more detail, I want to get more granularity, so I can search based on all kinds of detailed components. I can search based on a SNP or on a gene expression level or something. Is that clear? Yeah. And then two last things is, I mentioned already the fact that some people had their in-house format, and we're encouraging them to use, to at least bring into tranSMART things with a standard format, if we can. So because otherwise we'd be developing things, but maybe we'd have to have kind of a blob type object or so that we consider also. Yeah. I mentioned also there are a few design elements like data modeling with performance in mind. I think that's pretty self-evident. Okay, that's a pretty long category. And we now move into the category which is, so again, an axis on our roadmap component being data sources and loading. I want to upload. I think a lot of the uploading is done manually. We had already a number of interesting presentations on that. What I saw there is that, what I see more and more is that tranSMART is like this data warehouse or MART, but it feeds off from some kind of a data management environment, typically a clinical data management environment of some kind. And so a question that is being asked, I think really was mentioning it, is if people are dealing with clinical data and typically in a context where in such context CDISC standards are being used, if you could assume that the clinical data management environment takes care of curation and standardization, for instance, in a CDISC format, the next step could be to say, well, maybe I could simplify or standardize or automate the upload part. So I'm giving different steps of going towards a more streamlined way of uploading data. And then underneath, it talks about maybe we can refine the upload mechanisms. For instance, Stuyft automated lifecycle-based updating of tranSMART with newly available additional information, longitudinal data. I have a study, and it's not like everything is up to be uploaded, but I upload gradually. As time goes by, there's more information that comes. Imagine that you want people to work with tranSMART, but in that clinical data database, you have all your data, and only every 6 months tranSMART is updated. Nobody is going to go to tranSMART for a longitudinal study. So if you have longitudinal studies and you want to expose people to the data in an integrated way, you would want to have automation and kind of automated updating. Yeah, sometimes, for instance, one thing that I saw in OncoTrack also is that they say, well, we're not necessarily interested in the DNA constitution of one person, but we want to have the difference between the DNA of tumor and non-tumor. So you're starting to do calculations. And there I could imagine that this calculation you do outside of tranSMART, and then you feed it back with an update loop into tranSMART. And then there was also the question of, so now it is so, if you want to update some stuff, you have to get everything out and everything back in, apparently. So it's not like you have a lot of degrees of freedom. If you make a mistake with something, also correcting it. So these are, again, a number of things that were flagged up that I thought, is that important? And does this cover the main scenarios or not? Questions on this?
I have a question.
Yeah.
So when you use these schemas, so they have a very, let's say, fixed ontology scheme.
Yes.
What happens if you describe a loading procedure for tranSMART that's not compatible with the new version of the ontology, for instance?
In fact, this is a good question. First of all, I don't know if CDISC is the right thing to do, but indeed, whatever standard you follow and you have a problem there.
Because it's two layers, you know. You have like one standard that's going to be described in tranSMART to bits, and then come with the data in top of the standard. So then we got two problems of updates: the data of the standard itself and the data of the data that you want to load.
So what I would suggest is, if that is a topic that interests you, maybe it's something to delve into a little bit with the group, without losing the picture, the larger picture in view. I suppose that version control is a key element.
In view of time, I think we shouldn't do the discussion right now.
Yes.
And so
I suppose it wasn't the point, sorry.
Yeah, I mean, with this, obviously, it's going to depend on some of the standards are generic and some are disease specific. I think when you've updated something, you have to label and flag what version has gone in, when it clicks in. But all these things are looking for files, shouldn't they, in history of what's been done and how it's done. And then if we're cross-correlating with obviously another set of data, then knowing what version that's gone in and how that's done would be essential. So I think this is a common sense approach in most sense. Obviously, see this give you advice on the specifics when you're uploading these, and give you advice so you've gone off the line, and obviously give you pointers to the most relevant and updated standard. I think we then have to look at what's gone in already and make sure that when that standard is available, people are flagged that something is new and available. And then we look at updating that data. And doing that might be a bit of a trick. It might be ineffective to do touch and overwrite all the data again and to upgrade it. So the standards themselves, hopefully, are sort of back to compatible anyway. But I'll come back on that point because it will raise interesting questions as we're doing this at such a large scale.
Should we address all the rest of the questions as in the breakouts and in the discussions to try and get this?
Okay, so we come to a next section. We've had the modeling, we've had the uploading, and now we come into data browsing and querying. You want to make a selection of your data somehow, and with that selection, you want to do things. But first about the selection, we've talked quite a bit about the cohort selection. So if people here, again, I put a 0 saying the cohort selection GUI as it is in a number of cases enough, but in a number of cases one wants to extend the GUI paradigm. So for instance, to extend the diversity of selectable data elements. For instance, as new multidimensional data get into the whole picture, we'd like to extend the GUI so that we can pick up and build up queries with these. A structure is created into lower levels of detail. So, for instance, we want to have, for instance, DNA data, being able to say, I want to select these, maybe to export them as a whole for a certain biosample or a selection of biosamples. That's this here. Or I want to be able to go in more detail and say I want to capture that variant. Show me all the patients or all the biosamples where that variant, DNA variant, is represented. And then a next step could be, so this one could be, you have the current paradigm, but you add elements to it. The other could be the aspect like, for instance, the faceted search, where you make the querying more intuitive. And again, if that's not needed, it's not necessary to put an accent on it. But if you feel like that kind of faceted querying that is possible in other sections, should we also have that in the Data Explorer? That could be one here. Yes, another one. So here we are talking always about a GUI paradigm, but here I'm looking at, shouldn't we make the querying capabilities accessible to modules outside of tranSMART, like where you basically say, I make my data, taking into account some security, and we'll come on that, I make my data queryable interoperably by other tools. Yes? So that's that one. And then, yes, and then extend the security model. For instance, in project HBV here, they have this thing where they say, okay, we do an experiment, we do a clinical trial, observational. We look at individual patients and how they respond to standard treatment. But with the blood samples, we're going to do in vitro, we're going to do a number of treatments. And by the way, some of these treatments are things that we want to have our partners see, and some others we don't want them to see it. So that's again the security model, where you say different research partners might want to be able to see different things. So here again, that's one line, but that can cover the whole world again. And then the last one that I see here is that was one that Chris Marshall brought up, is, and I think it was also mentioned here a few times, to extend with federated search, to be able to transparently search for data across different tranSMART instances. So again, that's one. Yeah. So where you say I have a tranSMART instance here, OncoTrack, I have ABI-RISK, and after a while you say, well, we want to do cross tranSMART searches. That's the federated search. I mean, that federated capability. Any questions on this? No? Then we go to the last, but it's the biggest section here. See? So then you have your data modeled, you have them imported, you have made a selection of data, and then you want to use these data for analysis, visualization, and different things. And again here, we know that tranSMART has a number of functions that you can immediately use, and we've seen some of them this morning, but that ends relatively quickly. So one of the first things that people have asked again in OncoTrack also is to say, well, what we can't analyze in tranSMART, we would like to be able to make a selection of and then get an export of. So where you basically say, I have a selection, and please export everything that interests me for that selection, being a CSV file of some kind where you capture for every biosample what clinical data you have available in that clinical tree, and then another where you say, okay, I have DNA data, and where you export these DNA data to some folder structure, and you have some references in your first file. Yeah. So basically, the idea where you say, I do a query, I can't do anything with tranSMART analysis-wise that I want for this type of thing. Okay, for this selection, please export everything in a format that other tooling can work with it. So that's number one. Number two is where you say, well, maybe I don't want to export. Maybe I want to make tranSMART queryable by external tools. And for instance, we have this, I put in here an example of such functionality on the open source R in tranSMART. as you heard, because we don't have these I2B2 kind of APIs, what you do is you kind of have a leak security-wise. So for instance, in the case of OncoTrack, we said that's not what you want. What you want is kind of access via an API layer that shields off functionality from underlying complexity and refactoring. So if you have an API layer, whatever changes underneath in the technicalities you will not be affected by it, because you basically say, what I look for and how you do it, I don't care, but this is what I look for. That's what an API is about. You kind of have a contract about, I look for this and you give me this back. And how you do it, I don't care. But in this API, we also expect the security requirements to be covered, so that if an API is being called, that only those data that are allowed to be taken back are being taken back. So again, going from a simple canned analysis functions in tranSMART, we can't do everything, so we export it, to more refined. We start sharing objects and object elements with external modules, so they can do things with that. And then we come to a stage where you say, well, but if we can exchange information with tools of interest, which are these tools of interest? And we've had a few mentioned, like the ones that I heard mentioned in OncoTrack, but also today and yesterday were, so for instance, if you want to focus around visualization, yes, why not have an interface with Spotfire, where you basically allow Spotfire to query what's relevant from tranSMART, and have Spotfire, for instance, what really showed yesterday, I think, is what they call Spotfire guided analytics, where you have, on the one hand, a data analyst who is very IT savvy and who prepares something, but in such a mold that the biologist, who doesn't know all these technicalities, has an interface that works for him. Yeah. The other thing is an interface with R, where we have some of that in tranSMART, but where you would say, okay, I want to allow applications like R to query things, to make things accessible, to do calculations and so on. Yes?
Can you guarantee with this APIs the security?
How exactly, I don't know, but I know that in APIs that is often part of the functionality of an API to see, okay, this request comes from, do I have the right user? And
API keys is installed, so you
yeah.
register yourself as a user.
Yeah.
You get a key for someone to do.
Yeah. Yeah. Okay.
Yeah, there is a yeah, exactly. Exactly.
Yeah. Okay. So also, for instance, another aspect around visualization is the aspect of DNA data. So at J&J we have at some point, but it was broken, implemented the IGV viewer. That's a licensed viewer. We have proposed to the OncoTrack people, would a standard viewer, would that be something you could use in your DNA data? Because they have now a specific way of looking at data. And so what we propose them, can you put your data in a standard format? We'll make it viewable by, for instance, a J browser or an IGV viewer. And if that is good enough, we don't have to start coding specific stuff for you, for visualizing things for you. So in this particular case here, a custom DNA variation parser might be something that we need for OncoTrack, depending on whether they, yes?
I'm trying to achieve. Yeah, I must have.
I hope not.
The line breaks are different.
Yeah, you know what, I passed it on, and apparently the formatting was
Yeah.
kind of messed up in the
It's printed double-sided.
I know. But also you see that the formatting was a little bit lost in the transition. Okay. So I go to the last, no, not the last, about that one. Yeah, there were some things, circle diagrams, I don't know, that keep my displays.
Circus.
What's that?
Circus, not circus.
Ah, circus. Okay.
It's where you the round where you see all of the genome and you see all the connections between the genome.
Okay. Yes, provide viewing paradigms. Yeah.
When you're talking about DNA viewers, what exactly do you mean? Is this sequence viewer, a site viewer, the one
Genome brands, like J brands.
And doesn't that cover the several things? Doesn't that cover, like, if you dig into detail, you probably see the sequence? Does that allow you also to see the genes and so on? I haven't looked into these things.
That's what genome browsers do. You can go right from the chromosome level, get right down to the sequence level.
Mm-hmm.
You can link out. You can provide URLs to the genome browser, or you can load your tracks in.
What would be nice is if you provide something like a bed track, so you can actually see the positions in something like UCSC browser, which is very popular, or sample,
yeah.
And then let that link back to the tranSMART, which will also be take the data.
Yeah. So that discussion, if you could bring that in the group that fits, that would be very helpful. Because again, I'm picking up clues, but I'm not in depth enough in these things. Okay. So a last one about visualization would be like paradigms to view different things, different data types, multidimensional data. I mentioned the R, and then there are a few ones here also. Support for longitudinal subject study data. You remember we missed that time dimension, but also we had these different study subject types. I could imagine that if you would start to look for things that the visualization of the relationship between some data of a patient and some data of its xenograph or related in vitro data and so on is not self-evident. So I kind of capture that there. Support for that kind of visualization. Support for cross-study comparisons. And here I probably made the mistake to try to spell that out, and I probably am fully wrong. Here I'd say maybe we shouldn't go into that detail. If the group has questions, they can come to me and probably swap me and say you don't know it. Support for variations in analysis and visualization needs between different subject types. No, this is the same. No, this is longitudinal and this is the subject type. So longitudinal is where you say, I want to see over time certain things evolve. How do you visualize that? And the last one is, I want to see for different subject types, how does their information, their context, how does that build up and how does that compare? I understand that that could be very different. These trees could be very different from subject type to subject type. Okay. And
Emmanuel, is it okay if people who believe they know a better way to write a definition is just replace what you've put down there?
Certainly, certainly, yes. So if as a group you say this makes no sense, we replace it by something that makes more sense, no problem. Okay, and now we come to a last part, which I don't see quite as part of tranSMART, but kind of the what you do, the ecosystem around it. I called it a complementary research project space to support data analysis and analysis results capture. And I captured a few things, and again, it's not complete, but maybe it can help trigger some discussion. So what I see here is, and so typically what you see is you have your clinical data management environment, and then what you have is you start doing processing. Processing and reprocessing or analyzing is kind of sometimes in the same space. So what you have data, and you want to explore what can I do with these data. And typically what we see our data analysts do is they kind of scout the internet, they scout academia and say, hey, this one has a very interesting algorithm, let's try it. Let's try this. So it's very exploratory research where you take a data set out of tranSMART, take some tooling, mix them together and get something out and say, well, it doesn't work, and start over again. In fact, I'm talking here a space to support that type of thing. And then in the next stage, what happens is these researchers will say, hey, if we combine this and this and this together, that makes like a very valuable pipeline, and you get a space for more, I call it, repeatable research paradigms. And for instance, a galaxy environment could be something that you plug in there. But again, you're not necessarily in tranSMART. You're in a space where you do a number of things. But whatever you get out of that space, when it makes sense, you would want to re-inject it. Like, imagine you've done some calculations on some data, and you say, hey, that's very interesting. How can I get it back into the data warehouse? Because that's very interesting information. Or I do an analysis, and I say, well, that analysis report, I'd like to put it into some place that I meta data tag. For instance, in the search part of tranSMART. So there I see a link. I see like you have kind of an ecosystem where you do all kinds of things, and what you get out, you try to feed it back into tranSMART because it's data and it's content that is relevant for you. Does that make sense? Yeah, I suppose so, yes. And then the question is, one of the things that I noticed is that people have said, we want this, we want this. I don't see that as tranSMART, but I see it as something that in some way will need to or would benefit by interacting with tranSMART, even if it was to get data out and to feed back data in at a later stage. So that's that. And then I have a last set of ones that have relations with security. What I picked up related to privacy and so on is, given the fact that we get more and more, we talked yesterday about handprints. In fact, fingerprint information is very private information. And as we are getting richer and richer fingerprint-like information of people, we see that the algorithms of what is it, anonymization don't seem to work anymore. We see more and more in literature people being identified where they were supposed to not be identified. And so that raises a whole question of security, where you basically say, okay, if you are in that space, should we at some point, and is that part of tranSMART or not, I don't really care, and that's something people need to define. But if you say we make tranSMART available so people can dig out data, in a sense, that environment where you dig out data needs to respond to some security requirements, and that's what's in here. Like, you could go and say, to what degree you could do that procedurally, but could do that also more systematically, to say, well, if I do, should I, you know, in the context of privacy, the purpose, the limitation of purpose, where you say I can only get to data if I have a specific scientific question. Yes?
I suppose that we split security versus privacy, because privacy is a completely different subject compared to security.
Yeah.
Otherwise, we're going to mix it and
yeah.
completely different discussion.
Yeah. So what I wanted to put here is put above the water level a little bit some of these aspects. But if you find they go too far, we can just drop them also. Yeah. My main concern was here, if you start getting things out of tranSMART and start doing with it, what are your degrees of freedom there? And that's something that I try to cover here, but maybe it leads us too far. And if it does lead us too far, we could just skip that section. Yeah. So what I propose now, I think we're through. I would propose that we have 3 groups. So we've had the section about the data modeling. So if we go back to this one here. So we had this section, supported data types. It's one group that I'd like to come together and like to kind of evenly spread. So we're 2, 4, 6, 8. We have, how many people do we have? 17. 17, okay. 6 people per group. Yeah, excluding or including. Excluding, yeah, it's better. So Ioannis and I will kind of go between the groups. But so if you could split up in 3 groups. So one group is going to do the data, the modeling, so supported data types. The other group, group 2, will do data upload and data select, so data sources and loading and data browsing and querying. These are two small areas. And then the last one, facilitating data usage, so data analysis and visualization, is the third group. And so, is the assignment clear? You, as a group, you try to discuss how would we change this, the straw man, so that at the end of the day you can hand back an electronic copy of how you would see it. So I